All right. In this particular video, we are going to talk about the various organs of the lymphatic system, the general roles that they play, and just and how they kind of contribute to the overall immune response. Because I remember when I was a kid and I was learning about biology and a little bit about human biology growing up. I remember when the immune system was actually talked about as a completely separate organ system, and then after a while, rightfully so, I believe, people started to realize that pretty much most of the cells of the immune system that carry out specific immunity are stored within lymphatic organs. So the two were just kind of lumped together. It's imperative that you have a good general understanding of the organs of the lymphatic system and how the cells are organized within them in order to truly understand the immune response and how it works. So basically what we're going to do is we're going to just give a brief overview of the functions of the lymphatic system and then talk about some of the cells and organs and the general structure. And then probably in this video we will talk about the thymus gland as well. And basically because it functions differently than the rest of these organs, and I'll explain that in a little bit. So these, the lymph nodes, spleen, and tonsils, and Peyer's patches will be in another video. So on that note, let's kind of dive in. The lymphatic system. The lymphatic system serves two main purposes. One is to maintain fluid balance. And just to review, remember that the lymphatic system basically is draining, I shouldn't say draining necessarily, but basically extracting excessive interstitial fluid out of tissues. The lymphatic capillaries pull out excessive fluid buildup in our tissue fluids, interstitial fluid, that's what that is. And then basically that lymph, once that fluid is absorbed into the lymphatic system, we call it a lymph, and then it is circulated throughout the lymphatic system, and it's eventually recycled back into the bloodstream, thus basically completing the fluid cycle. Because remember when you were learning about capillary fluid dynamics and you were learning about filtration on the arterial end of capillary beds, reabsorption on the venous end, and remember we said that only 90% of fluids that are filtered out of capillaries are reabsorbed back into the bloodstream directly. So that means 10% of fluid on average that we filter out under kind of normal resting conditions remains within the tissue spaces, and over time that could accumulate and cause edema. However, the lymphatic system is there to grab and drain any excessive fluids out of our tissues and, like I said, return that extra 10% of fluid back into the subclavian veins, i.e. the bloodstream, thus, like I said, completing the fluid cycle and maintaining fluid balance. And then, as we've already discussed and you've already learned about before coming to this point, excuse me, there are, basically there are ways we can increase, the circulation of lymph, increasing cardiac output, increasing blood flow to our tissues, increasing filtration. That's going to increase the flow of lymph and so on, like during exercise and whatnot. But in general, that's kind of that's one of the main functions of this. But like I said, we've already kind of covered fluids and whatnot, so I'm going to kind of leave that be because the main topic that I want to focus on through these next few videos is the immune system aspect of the lymphatic system. So basically, the lymphatic system, or the organs of the lymphatic system, are responsible for the maturation and the storage of cells of the immune system. All right. So basically, cells are, like I said, blood. Remember, the immune system is composed of blood cells. To be more specific, white blood cells. So remember that all blood cells are created in bone marrow. Remember, they start out as the stem cells, and then they are later on converted into specific white blood cells based on different types of chemical messages that they interact with, essentially. But then they are produced in bone marrow, and then some of them are matured in bone marrow, some are matured in other glands, which I'll talk about more specifically in a bit. But basically, once cells of the immune system are matured, then they are stored inside of our lymphoid organs, your lymph nodes, your spleen, tonsils, Peyer's patches, and so on, They are stored in there and just waiting to come in contact with a pathogen. All right, and we'll talk about that process more. So maturation and storage of cells of the immune system. And one thing to remember is that these are like any other organ; they are capsulated. So basically, what I'm saying here is they've got an outer connective tissue border, and that's kind of one of the structural differences between lymphatic organs based on their location. Some of these capsules are more thicker, more thin, and but again, that's going to play a very important role into their function in the first place, which I'll dive into later on. All right. Now, if there's one common feature that of the organs of the lymphatic system share is that they are all filters minus one, basically, with the exception of the thymus gland. So lymph nodes filter lymph, the spleen filters blood, tonsils filter whatever's blown in or through our nose and our mouth or whatever we consume. Peyer's patches basically filter bacteria that came in from the GI tract, that we ingested, or or any bacteria that may have stuck out of the colon into the small intestine. All right, but in general, all these lymphatic organs act as filters. Like I said, with the exception of the thymus gland plays a critical role in the maturation of T cells. Again, I'll focus on that later. All right, but in a nutshell, when we think about this, basically being that these are storage facilities for cells of the immune system, all right, that basically means their primary responsibility is carrying out the immune response. And a big part of carrying out an immune response is developing a memory to pathogens that we are exposed to. But I'll tell you right now that we don't essentially develop memory until we are exposed to pathogens. I'll explain that more in a little bit. But that's just kind of something to keep in mind. So remember, I said that all but one of these organs act as filters. So basically, we pump some kind of fluid or push something into or through these lymph nodes, and whatever goes through or comes in contact, not lymph nodes, I'm sorry, lymphoid organs, whatever comes in contact with these organs essentially is going to come in contact with white blood cells that are stored in there. And as a result, if they are a pathogen, we will carry out an immune response against them, and then we will develop a memory against them, so we can carry out a more effective response if we are ever exposed again. All right, but in a very general sense, that's basically what the lymphatic system does. Now, let's kind of think about this a little more specifically in terms of functionality. When it comes to classifying organs of the lymphatic system, we can break these down into primary and secondary organs. Okay, primary and secondary organs. Primary organs are, this is where the maturation process comes in. Okay, the maturation process comes in. All right, so basically, I'll tell you right now that the two major cells that carry out the specific immune response are T and B cells, or more specifically, T and B lymphocytes. All right. Now, basically, the reason why we keep saying that this is specific immunity is because T and B cells react to specific antigens. And remember to review, an antigen is a unique marker. It's a unique molecule, essentially, found on the, or molecules found on the surfaces of cells. All right, so basically, they're these unique markers found on surfaces of cells. And every living entity, every living cell essentially has some kind of unique array of molecules that the immune system can recognize. All right. And basically, if the immune system recognizes these as foreign, basically foreign meaning not originating in us, then the immune system will react to it. And basically, when the immune system reacts, or essentially is activated, it attacks. And it attacks for the purpose of eradication. And basically, the immune system doesn't stop reacting until that antigen is gone. Now, because this is such a specific type of response, because the cells are reacting specifically to just surface molecules, when we carry out a very specific response like this, we are only going to attack those cells that carry those specific antigens. All right, so that's a very, very precision type attack versus like a physiologic response, like a fever that affects the entire body. All right, that can cause complications everywhere in the human body. All right, when we carry out an immune response, like I said, it's very, very precision. We're only targeting specific cells that have these antigens. However, that can be bad sometimes because Self-antigens, these are, remember I said essentially every cell, I mean including viruses, have specific molecules on their surface that are essentially identifying molecules or identifying markers. All right. And that includes the cells of our own body. Now, basically, when it comes to self-antigens, these are antigens that originate within us. All right. So obviously, we clearly don't want to react to these, because if we react to these, that means our immune system is essentially going to attack our cells. We're going to attack our own tissues. And last time I checked, that's kind of a no-no. That basically is an autoimmune disease, when the immune system reacts to self-antigens and targets that specific tissue and goes after them. All right. So that's kind of something to keep in mind. So basically, primary lymphatic organs mature T and B cells. So basically, when we say they're mature, we would refer to them as immunocompetent. All right, we would refer to them as immunocompetent. So an immunocompetent T and B cell, a mature T and B cell, knows the difference between a foreign versus a self-antigen. Okay, mature immunocompetent T and B cells do not react to self-antigens. They leave them be. All right. However, they, on the other hand, react to foreign antigens. So you're probably wondering, well, how does our body know this? Well, one of the ways, basically, as we are undergoing embryonic development, basically our cells of our immune system are going around and basically coming in contact with all the cells of our, not all, but most cells, because there are some tissues that develop faster than the immune system can get to them. Like, for example, the proteins in the lens of your eye. Those are molecules that your immune system has never seen before. So if your immune system were to ever come in contact with them, it could damage or destroy them. All right. Gametes are another example, like sperm. I mean, there's a reason why sperm and blood are separated from one another. So there really isn't direct blood flow directly into the areas of the testes where sperm are matured and manufactured, because we would kill them, because you don't start seeing sperm development until after puberty. So basically, during embryonic development, our immune system is surveying our tissues, and basically by the time we're born, our immune system has basically seen all it can see, and that's how the immune system knows to leave our own antigens alone. So anything that basically our immune system comes in contact with after we're born that it's never seen, we're going to react to it. And like I said, that includes a cell like spermatozoa. All right. So that's something to keep in mind. So basically, the thymus gland is the area where T cells mature. All right, this is where T cells. Now think about that. Thymus. Thymus starts with the letter T. That's essentially why T cells are called T cells. Okay. I'll explain this process more in depth, in extreme depth, but I'll explain it a little more in a little bit. But basically, this is where T cells become, when I say they become mature, they become immunocompetent. This is essentially where T cells learn how to tell the difference between a foreign and a self-antigen, all right? And if T cells cannot do this, then they are essentially destroyed, which I'll explain more in a bit, all right? Now, so obviously, process of elimination would leave red bone marrow as the site where B cells are put to the test, where B cells are essentially mature or become immunocompetent. Now, again, you're going to want to think that, because bone starts with B, that's why B cells are called that. That's actually not true, because when this was first being studied, this was actually studied in birds, and birds have a little area, kind of like a little sac down by their rectum called the bursa of Fabricius. The bursa of Fabricius. Luckily for us, bursa also starts with the letter B. All right, but basically the bursa of Fabricius in a lot of birds is essentially where B cells undergo this process of becoming immunocompetent. And then, like I said, it's just coincidental that bone marrow starts with a B and bursa starts with a B, so we just call them B cells. All right. So basically, so that's something that's a little goofy though, if you think about it. T cells are produced in red bone marrow like any other blood cell, or any, like I said, red blood cells, white blood cells, platelets are all manufactured in red bone marrow. However, and by the time blood cells leave bone marrow, they're under normal conditions, they're fully functional, ready to go. Neutrophils, eosinophils, basophils, monocytes, erythrocytes. Platelets aren't actually cells; they're fragments of cells. Whereas T cells are kind of the goofy ones here, because when we produce T cells, they actually leave the bone marrow in a very underdeveloped or immature state, and then they go to the thymus gland where they learn how to become immunocompetent. And if they don't pass the test, then they fail. All right. And then basically once T and B cells become immunocompetent, then they are basically circulated out of bone marrow and the spleen, and then they are sent to basically these lymphoid organs, or these secondary organs, to be stored. All right. And basically remember that all of these structures are essentially filters. All right, Peyer's patches are a cluster of filters found in the distal end of the small intestine. Tonsils, these are filters that are found in the nose and the mouth and throat, essentially. Lymph nodes are basically connected to basically lymphatic vessels, and lymph is pumped through them. The spleen is connected to the bloodstream via the splenic artery. All right, so basically whatever materials are sent to these structures is basically going to come in contact with the T and B cells that are stored within them. And if there's any pathogens in that material that's sent to these secondary organs, then T and B cells are essentially going to react to them and will carry out an immune response. That essentially is how the immune system works, again, on a very general scale. And then that's the purpose that primary and secondary lymphatic organs serve. All right. Now, when thinking about the general structure of these lymphatic organs, basically, again, there are some commonalities that they share. For example, there's stroma. Basically, stroma is kind of the way of saying that's kind of the histology term for the kind of like the internal skeleton of an organ, the internal framework of an organ. So basically, it's the like I said, it's like the internal skeleton. Basically, it's all the different protein fibers in the extracellular matrix of that tissue, and they arrange in a specific fashion to create kind of, like I said, like an inner scaffolding or inner framework that gives that organ structure. And obviously, that structure plays a role in the functionality of that organ. Okay, and when we're thinking about these lymphoid tissues, the one thing that they all share is that they are made out of, or that internal framework essentially is made out of reticular connective tissue. Now, reticular connective tissue is very soft, okay? Because reticular connective tissue is going to sound kind of goofy, is made out of collagen fibers, okay? Now, I know if you've ever listened to any of my other videos on histology, or taken any of my classes, you'll always hear me talk about collagen as a really tough structural protein, all right, which it is. However, this has to do with the design of the collagen, essentially these reticular fibers. Basically, reticular fibers are very short, and they kind of have branches. So basically, they don't cover a lot of area, and they create a whole lot of space, all right? So reticular connective tissue, reticular fibers, the framework that they create essentially creates a lot of open spaces, but that's a good thing because we need a lot of room to pack all these white blood cells into these lymphoid organs, all right? So like I said, reticular connective tissue creates room to store these organs. So because the spaces between this type of tissue is so widely spaced, that makes lymphoid organs very delicate. And if you damage them, I mean, you're probably going to permanently damage them. Like, for example, when somebody ruptures their spleen, if you try to repair it, the odds are you'll probably wind up doing more damage to it just trying to repair it because of how soft and delicate it is. All right, so that's kind of something to keep in mind. All right, so basically all these organs are their internal framework, their internal stroma is made out of reticular connective tissue, a widely spaced form of connective tissue that, again, that creates a lot of space, that creates a soft inner skeleton, but allows for a lot of room. And like I said, if we're trying to densely populate this tissue, this organ essentially, with a lot of white blood cells, that's the kind of inner framework that we want. All right. Another thing that these organs share in common structurally is that they are all encapsulated. So they all have some kind of outer connective tissue capsule. All right. Um, Now, the thickness of that capsule is going to vary. For example, the spleen has a pretty thick outer capsule. All right, lymph nodes for their size have a pretty thick outer capsule. All right, tonsils, on the other hand, tonsils in these Peyer's patches have a little bit thinner capsules, but I'll explain why that's important in the next video. But again, that plays a role in their filtering, in essentially how they perform their filtering functions. All right, but regardless of the thickness, how thick or thin the capsules are, they all are encapsulated. All right, and then obviously all these lymphoid organs are densely populated with white blood cells. All right, not just T and B cells, but also phagocytes, which I'll explain in a little bit. All right, now typically the variation in structure a lot of times depends on their location, because for example. Tonsils have really thin outer capsules. Tonsils have really thin outer capsules. Now think about that versus, like I said, the spleen, which has a really thick capsule that surrounds it. Okay, but let's think about this for a second. Tonsils are located in the nasal pharynx. They're located in in the palate. In the roof of the mouth, lingually in the tongue. All right, basically there's not a lot of material circulating into them via, the bloodstream. Lymph nodes have this fluid being pumped into them. The spleen has blood being pumped into it, whereas tonsils are coming in contact with materials on a physical basis. So basically, we breathe air in, or we consume materials, food and water. And basically, having that thin outer membrane makes it easier for materials to diffuse in and then come in contact with the white blood cells inside. Okay, so that's kind of something to keep in mind, and I'll talk about structural differences a little more specifically as I get to them. And again, cells of the lymphatic system, the lymphocytes are T cells and B cells. T lymphocytes or B lymphocytes. I'm just going to refer to them as T cells and B cells, but just remember that they're lymphocytes, and remember that lymphocytes are cells that have basically, they have a very, very large nucleus. The nucleus practically takes up the entire inner surface area of the cell. So when you look at one of these under a microscope, you're pretty much just going to see a big nucleus and a little bit of cytoplasm, and very, very incredibly fine little kind of grainy granules. In the cytoplasm. All right. Now, when an immunocompetent T or a B cell is in an inactive state, you're really not going to be able to tell the difference between the two just by looking at them through a microscope. You really start to see the differences between these cells when they are switched on or activated during the immune response, and then they take on very different appearances and functions, but that's something that will be saved for when we get to the immune response itself. And then there's another kind of lymphocyte that's not nearly as abundant, but it's important, called natural killer cells. These are often nicknamed surveillance cells. So these are cells that kind of wander around, within lymph nodes and kind of around the body. And basically what they're doing is they are just surveying our body tissues. They're just sitting there looking for problems, looking for foreign antigens, and then reacting to them and killing them. It's kind of cool. What natural killer cells do is they get really close to a cell. They release proteins that basically punch a hole in the cell membrane of what they're trying to attack, and then they release enzymes that basically digest that target cell from the inside out. And basically those lytic enzymes are going to cause so much damage inside of that cell that they're going to induce apoptosis, basically program cell death, and which is kind of a neat little feature. But those are natural killer cells. Now, one of the other more important cells that we find stored inside of these lymphatic organs are phagocytes. All right, phagocytes, like macrophages and so on. Now, one thing that's really important about these is that you understand what phagocytes do. Phagocytes eat things, right? So let's say here's a bacterium. A phagocyte's gonna come up to that bacterium, and remember, you guys remember how phagocytosis works. Basically, with phagocytosis, that cell is going to create kind of an involution of its cell membrane, like an inward folding of the cell membrane, and it's going to literally engulf the material that it wants to phagocytize and create what's called a phagosome. And then basically that phagosome is going to, basically this little vesicle with the material it just ingested inside the cell is going to fuse with the lysosome, and those lytic enzymes are going to digest and break apart whatever this phagocyte consumed. However, many phagocytes, what they'll do, especially if they consume a pathogen, what they'll do is they'll take some of those proteins and they will stick it. They will stick it on the surface of themselves. So basically, they'll put this molecule on this essentially this pathogen on their surface, and that's really important because then what these this is why we call them antigen presenting cells. They are presenting an antigen. And essentially what they're going to do is they are going to play a critical role in activating T and B cells and switching on the immune response more rapidly. All right, so that's kind of something to keep in mind. And again, that's a topic I'm going to revisit later on. So let's talk a little bit about the thymus gland and the role that it plays. So like I said, the reason why I'm including the thymus gland in this video, then I'll talk about all the other ones separately, it's just because the thymus gland is different because its main job is to mature T cells, all right, whereas all the other organs act as filters. Now, the thymus gland is located, anatomically located in the mediastinum, all right, and remember, this isn't the first time. By now, if you're getting to the lymphatic system, this isn't going to be the first time you've ever heard this word. The mediastinum, remember, is the physical space between the lungs. Okay, it's that physical space. So on the medial side, the lungs do not touch each other. They do not come in contact with one another, and as a result, there's a space in between. All right, and basically within that space, that's where you find the trachea. Then the trachea bifurcates into the main stem bronchi. That's where the heart is located. That's where the aorta and the inferior superior vena cava are located. That's where the thoracic duct of your lymphatic system, passes through there. And obviously the thymus gland is located in there as well. Now, the thymus gland sits on the superior aspect of the heart. So the thymus gland is superior to the heart. Okay, and remember your anatomic terminology, 101. Remember, superior means above or towards the head. For this sake, we'll say above. So basically, the thymus gland sits above the heart, but it's below the thyroid gland. So I always like to think of the thymus gland as kind of being in between the two. All right. Now, basically, what happens here is the thymus gland puts T cells through a test, basically through positive and negative selection, which I'll talk more about in a little bit. But basically, this is about essentially making cells immunocompetent. Through positive selection, this is basically when T cells develop their receptors called MHC receptors, major histocompatibility complexes. And then negative selection, this is a test that they go through to make sure that they don't recognize self-antigens and react to them. And if they fail any of these tests, they're basically going to be eliminated, which I'll talk more about in a second. All right. Now, there's a couple of things that I want to talk about related to the thymus gland as well. The thymus gland basically undergoes some unique changes throughout our lifespan. So when we're first born, the thymus gland is quite large. It's actually very, very large. The thymus gland is actually a hair bigger than the heart when we're first born. All right. So basically, you've got your heart, and then you've got a big thymus gland kind of sitting above it. All right. And then basically, it will grow slightly throughout childhood. All right. It will grow a little bit as we get older. And then eventually, once we kind of start getting into early adult, kind of late teens, early adulthood, the thymus gland is actually going to start to atrophy. It's going to shrink in size. All right. Now, this is an interesting little phenomenon. So basically what's going to happen is once the thymus gland starts shrinking in early adulthood, it's basically going to shrink in size on average about 3% per year. So what I'm saying is that every year as we age or as we get older in life, like I said, think in early adulthood and on, the thymus gland is going to be 3% smaller every year. All right, and then eventually, what happens is that the thymus gland, as it shrinks, the functional parts of the thymus gland as it shrinks are replaced by adipose tissue or fat, and then eventually, once you start getting into your mid 40s, early 50s, the thymus gland is essentially the functional thymus gland, I should say, is essentially gone. Now, so basically, it doesn't just disappear. Like I said, it's basically replaced by adipose tissue. So if you were to look at a cadaver of an elderly individual, basically what you're going to see in the area above the heart where the thymus gland used to be, you're essentially just going to see kind of a chunk of fat. All right. Now there still needs to be some research that goes into the cause of the shrinkage, or the appropriate term for this is the involution. Okay, the involution of the thymus gland. Now, basically, with this, one of the kind of the leading ideas out there as to the causes of the involution of the thymus gland is they believe that that there's a correlation between as we increase our sex hormone levels, that those have an effect on the thymus gland and cause it to slowly shrink in size over the years until the point where it's eventually gone. So it's kind of just something to think about, but again, a lot of that still needs to be researched. Now, however, there's obviously some implications to that, because I mean, think about that. Remember, the function of the thymus gland. This is where T cells are matured. All right, this is where T cells are matured. Now, think about why it's so big when you're a kid. Think about why the thymus gland is so large when you're a kid. I mean, remember, I said it earlier, and I'll reiterate it again. We develop our immune system by exposure. We develop our immunity by exposing ourselves to pathogens. And when we expose ourselves to pathogens, that's when T and B cells are activated. And when I say activated, they're switched on and they carry out an immune response. And part of that process is we develop these genetically programmed memory cells. So if we're ever exposed to that specific pathogen again, we are going to kill it, or at least fight, or at least do our best to kill it a lot more quickly. What was I going to say here? Now, basically, so think about that. I mean, when we're first born, having that thymus gland so big, it plays a critical role in developing and maturing T cells as we're growing in life. All right, and then eventually, like I said, as we get older, that's gonna shrink down, and then eventually, what's gonna happen is it's gonna disappear, and then that's one of the reasons why this has obviously been linked to a drop in the functionality of your immune system as you get older, and this is this is why they believe that as we get older, that we're more prone to certain types of infections. And then cancer is more prevalent in the elderly because T cells carry out something called cellular immunity or cell-mediated immunity. Basically, T cells attack cells. They attack bad cells, essentially, cells with foreign antigens on them. All right. Now, luckily, one of the nice features of T cells, though, and B cells, is that they tend to live for a long time, especially memory cells. So just because you lose your thymus gland doesn't necessarily mean you lose all functional aspects of T cells in your immune system, because you still have a lot of memory cells left in you. It's just that you're just going to have a harder time, obviously, developing, or maturing more, newer T cells without the thymus gland itself. So you're kind of banking on kind of an old, aged immune system. All right. Now, so let's kind of take a look at, again, review the anatomic location of the thymus gland here. So basically, remember, it's in the mediastinum. So you've got your heart. Hopefully your heart doesn't look like a caiman's head. And then basically you've got your lungs. Forgive my art, people. Even if I don't have this tablet, I tend to draw at a fifth grade level, at least here. I can blame the stinking tablet. And apparently your lungs are going to be disproportionately sized, but you get the picture. Basically, that was a terrible drawing. Actually, I should have drawn this a little more here. Sorry, you get the picture here. But basically, the thymus gland is located in the mediastinum, the physical space in between the lungs. And like I said, it sits atop the heart. It's superior to the heart. And then normally, like I said, your thyroid gland is going to be right around this area here. So like I said, the thymus gland is in between the thyroid gland and the heart. All right, and remember that word. I can't emphasize that word enough. Mediastinum. Mediastinum, because like I said, that's just such a busy area in the chest. That's just an area. That's just something you're going to encounter a lot. So keep that in mind. All right, and then internally, when we take a look at the thymus, basically again, its inner stroma or framework is composed of reticular connective tissue, so reticular CT. Now let's kind of talk about the functionality of this again for a second. So when we look at the kind of the functional inner aspect of this, basically this is where structurally the thymus gland is similar to the other lymphoid organs, is in the fact that it has a cortex and a medulla. It's got an outer functional covering, and then a middle inner functional area. Now, the cortex, this is essentially where positive selection occurs, all right? So basically, we get these very, very underdeveloped or very immature T cells that get into the thymus gland, and they undergo positive selection. So once they get in there, basically, they kind of get exposed to all these chemical messages and cells in there. And then what they're doing is they start developing receptors, like remember those MHC receptors that I mentioned? These are receptors that are very, very specific to T cells that play a critical role in recognizing foreign antigens and activating the immune response and enhancing the immune response and carrying out immune functions. All right. So basically, because when these T cells first get in here, genetically, they're just kind of a mess inside. So what they start doing is the genes inside start to kind of arrange themselves, and then they start to form these receptors. And essentially, if these receptors develop properly, then the T cells migrate into the medulla of the thymus gland, where they undergo negative selection. Okay, so positive selection, we want to make sure they're positive for these receptors. They're essentially working properly. And then once they migrate into the very, very inner, the inner portions of the medulla, this is where they again start to undergo that negative selection. And basically what's going to happen here, there are epithelial cells antigen presenting cells, and there's just a bunch of cells inside of here that are basically going to make these T cells come in contact with various antigens. However, the antigens that these developing T cells are going to come in contact with are self-antigens, all right? And they're going to come in contact with just a wide variety of all of these different antigens. And if they react to any of these, that's bad, because that means they're reacting to self-antigens. That means these T cells would essentially want to carry out an attack on our own tissues. So basically, at any point in time, if T cells fail either of these tests, positive or negative selection, they're going to be eliminated. All right, basically, they're not going to be exposed to the right chemicals. All right, they're going to be neglected. They're going to be attacked and damaged and forced to undergo apoptosis. But in a nutshell, they're going to be eliminated, because we don't want a bunch of T cells going out there with non-functional receptors that aren't essentially going to react to antigens just because their surface proteins aren't working properly. And we certainly don't want T cells that are reactive to self-antigens, because then we'll start attacking our own tissues. So basically, we kill these off whenever they don't, essentially what I like to say, pass the test. Now, this is a pass-fail ordeal here. There's no B, C, or D grades in this scenario. It's either pass-fail. If at any point in time these maturing T lymphocytes fail this test, then they are eliminated. Now, this is kind of interesting, and this is good as well, that over 90% of the T cells that undergo this testing process, this maturation process, fail. Over 90% fail, but that is a good thing. All right, we want to be picky with this process. This is not something we want to do half-cocked. I was going to say half rear end of a donkey there instead, but you get the picture. But basically, again, this is a specific process, and it's good that so many of these T cells fail, because remember that specific immunity, whether we're talking T or B cells, this is about reacting to very specific proteins. All right, we want to make sure that these cells aren't going to be highly reactive to antigens or other molecules that could cause damage to our cells. And B lymphocytes undergo a similar process in red bone marrow as well. They're exposed to their surface antigens develop, and then they're exposed to just this myriad of self antigens as well. And if they show that they're reacting to these, then they are gotten rid of. Okay. So that and then I was going to say, and then when the T cells do pass this test, then that's when we refer to them as immuno. I'm just going to abbreviate that. I don't feel like writing the whole word on this tablet because you won't be able to read it anyway. But that essentially is what we refer to as immunocompetent. These cells are immunocompetent. They know the difference between a self and a foreign antigen. And then basically these cells are these newly developed immunocompetent cells are going to leave the thymus gland. They're going to get into the blood, and then they're going to be circulated into our lymphoid tissues where we're going to store them, lymph nodes, spleen, whatnot. All right. Gosh dang, there's something else I want to say about that too. What is it? Shoot. There's something else I want to say about that, and I forgot. It left my mind. Must not have been that important if I am not remembering it. My apologies, folks. But in a nutshell, that's essentially, the thymus gland, and you can see how that essentially relates to the functionality of the immune system. Now, like I said, this, in a nutshell, is how the immune system works. Remember, recognizing self versus foreign antigens. We need to, as we develop T and B cells that carry out specific immune responses, we need to make them immunocompetent. We need to make sure that when they come in contact with self antigens, that they are left alone. All right, and then once they do become immunocompetent, we circulate them to our secondary lymphatic organs where they are stored and just wait to come in contact with pathogens or foreign antigens so we can essentially kill them off. All right. So that is, and another thing I was going to mention too about the thymus gland, this is just more interesting than anything else, is that the thymus gland is often studied when thinking about the biology of aging and how changes in the thymus gland affect aging. What's interesting is that when we're born, if there's any problem, if the thymus gland is essentially removed after we're born, we'll still be semi all right. But if the thymus gland it doesn't develop or is removed, like in animal studies, or if it doesn't develop in us after we're born, then this is going to be problematic, because then you're not going to. Basically, it's been shown that you're pretty much not going to have really any T cells, mature. Naive T cells. Naive T cells are cells that have become immunocompetent but never seen an antigen. All right, you're not going to have any of these, and then it also causes just people, animals and people to just age really quickly as well. So, so the thymus gland is something that has been and still is being studied when it comes to thinking about the aging process as well. But there's still a whole lot that needs to be really understood about it. So. But that essentially is the thymus gland, and like I said, I lumped this in with this video just because it kind of ties into the functionality of the immune system in terms of developing immunocompetent cells. And in the next video, we're going to focus on the secondary lymphatic organs, the lymph nodes, the spleen, the Peyer's patches, and the tonsils.